they invade, and so somehow the cancers, they evade the growth suppressor so that they can continue their stuff. And then one of the property for this one is the activating invasion and metastasis. As you know, that is why it is malignant in nature. And then, and because they have they need to keep on proliferating, so they need to have the replicative immortality. What do you mean with the replicative immortality? The mean that they need to sustain. They need to sustain, and then they need to repair. They need to repair in the faster pace, because there will be a lot of endogenous damages happen when they replicate, because the rate of replication is very fast, and due to that, in cancer cells there are a lot of mismatch repair or repair aberrations are there. And then other is they induce angiogenesis. That's why they need to grow, and definitely they resisting the cell death, or you can say the apoptosis or metastasis. So based on that, people around the world or scientists, they have made a lot different inhibitors against each and every hallmark. EGFR signaling is there, EGFR emitter is there, which inhibit the growth proliferating signaling. They have like the cyclin dependent, the CDK kinase inhibitors, and the cell cycle. There are a lot of cell cycle inhibitors. So what they do, they try to stop the growth, and then they have the immune checkpoints. They have they can grow a lot, so they need the telomerase, like one of the stuff is the telomerase, but still there are a lot other inhibitors are there. And then they also have the anti-inflammatory drugs which can stop, or we can hinder the tumor progression. They have a metastasis inhibitor. They have like VEGFR, EGFR. There are a lot of signaling which can inhibit angiogenesis. And then we have also the genomic instability or mutations. You can say that here is the DNA repair, which is one of the example is SPARC. And then they have the apoptotic inhibitors, and then they have some sort of deregulation. And one of the example is methotrexate, which has been used as a inhibitors. So over the period, they have made a lot of, we have mentioned only 6, but there are thousands of inhibitors, both clinically and the preclinically, and in the early phase of development, they try to hammer the cancer with the different signaling pathway. So I would like to say something about it is very surprised to know that I don't know if somebody has an idea how many DNA repair, how many DNA lesions per cells per day. So here I can mention that there are two kinds of the DNA damage could happen. There could be the endogenous damage and could be exogenous damage. So if you can see, there are different damages are there. Some different lesions are there. It could be the double strand break. It could be the cytosine deamination, cyclopurine adduct, depyrimidation, 8-oxyguanine, alkylation, depurination, and single strand break. So you can see here, there are so many damages, both endogenous as well as from the environmental. Once we exposed like from the sunlight, from the background radiation, from IR or radiation therapy, from the chemotherapy. So one can say that we have something about 10,000 to 100,000 or more lesions per day in the cells. So how they survive? Because, and how they can cope up with such a massive DNA damage? Because they have very robust system to repair these lesions. I would like to stress upon one stuff about the Among all the lesions, so DNA strand break lesion is the most lethal. And we say that if the lesion is got around 40 double strand break in a cell is enough to trigger the apoptotic responses. So what is our objective with the DNA repair limiter? What we do any kind of damages that leads to form the single strand break or deamination or apiurine formations. Ultimately, what will happen in the over the replication stress, over the replication period, they will turn those lesions to this double strand break. And then if they are not repaired, and then there is some the massive mechanism to repair the double strand break. If they cannot repair the double strand break lesions, cells will be triggered for the apoptotic signal. So what is the source of, as you can see the previous slides, what is the source of the damage? So here is like a DNA replication stress. What is DNA replication stress? DNA replication stress is like anything which hinder the replication, which slow down the replication is called the DNA replication stress. It could be due to the single strand break or anything which can oxidative damage. It can say that this is the DNA replication stress. The free radicals or ROS, what we say, they modify a lot of nucleotides. So or the ionizing radiations, chemotherapy, they induce single strand break lesions. The polyaromatic hydrocarbons or UV lights, they induce some bulky lesions, some sort of DNA adducts. So they induce a lot of intra-strand or inter-strand cross-linking. Then IR or chemotherapy drugs, they induce double strand break. So we have a lot of different types of damages. The BER, base excision repair, they have single strand break repair, they have adduct, they have double strand break repair. And then what they have, the cells have very good efficient system to repair these damages. And this is called like, the mismatch repair, the base excision repair. And these we'll talk about a little bit more how the base excision repair is happening. And then we have also the interstrand and intrastrand cross-linking. So here, the nucleotide excision repair, they play the important role, and it could be the transcription associated or global genome repair. And then we have the double strand break repair, and it is mainly to the two different pathways, either homologous recombination and the non-homologous end joining. So you can see this cartoon or the picture that we have the different cancer type, like the radiotherapy or radiometric therapy. So when you do the radiometric therapy, so they induce some damage to the cancer cells, right? So they induce some toxic lesions to the cancer cells. Then how they repair? So here, as I mentioned, there are different repair pathways, and here in the space, in the particular on the radiation non-homologous end joint repair, they play very important role. So people can think that something, if we target the non-homologous end joining repair with the radiation, one can have the better effect. Same with the mononucleating agents. You can see that they induce the base damage. So here, both the base excision repair and HR. I mean, you can say in the mononucleating function, there are lot many repair pathway they are involved into repair the DNA. And then they have the bifunctioning alkylating agent, which is very common with the cisplatin, mitomycin C, nitrogen mustard. So they all induce the double strand break. And then we have the system, the homologous recombination and Fanconi anemia pathway, these, and then even the NER, especially for the cisplatin, they repair the damage. And then we have also the antimetabolites. They use the 5-FU, folate analogs, and they have not so many known, but they say that it's thought BER. Then we have the topoisomerase, camptothecin, adipocytes. There's also like we can say that, and we have different pathways involved based on the lesions. It is not that one lesions can be taken care of by the particular pathway. Then our interest is like homologous recombination, and I don't want to go in very detail how the homologous recombination happening, how the NHEJ happening, how is the but I will say a little bit which are the protein which is involved in the different kind of repairs. And here you can see that the basic strand repair, and I'm sure everybody's aware, like the single strand break repair, and then most of these things is taken care of by the OGG1. This is the DNA glycosylase, and then the PARP inhibitor. So what they do the PARP, because I will talk about the PARP inhibitor. So what the PARP inhibitor do? So what they do, they polymerize and then become PARP. And then once they have the PARP, and then they bind, because they have domain different kinds of, and they have the DNA binding domain, and then they bind to the damaged sites, and then they attract the different protein which is involved in XCC1, and then you can see that they cleave it, and then they have the polymerases, they have the antigen, PCNA, they have FEM, and then ligase. So these all the repair pathway, they are the complex, and definitely there are some sort of overlapping between the different pathways, and the XCC1, ERCC1, and then XPC. So like I can say that every repair process, they have the involvement of the polymerase, they have the involvement of the ligases, but definitely all the polymerases and ligases, they also have particular function. I mean, it's not that ligase 3 is work for that when ligase 1 is work for that. So they have the most of the protein, they have more specific function. And then similar, they have the mismatch repair, they involve in the Pol delta, the Pol, and then they have the ligases here. And then classical is like HR, the homologous recombination, where the BRCA2, I'm sure might have heard of the BRCA2 repair, because if the person is BRCA2 deficient, especially the females, and they see is likely to get some ovarian cancer as well as the breast cancer. And the homologous recombination or non-homologous in joining, so these are very complex. So here the pathways like, the ATM pathway, the ATR pathway, the MRN complex, and these complexes, these are the first guardian. So they sense the damages, and then they become the RPA1, and then RPA, they try to recruit the gamma H2X, the 53BP1, and these proteins, they have their own specific functions. They carry out the repair process. If they fail to carry out the repair process, there will be damages and cells will die. So that's why it is critical to hammer the DNA repair so that we can make the cancer cells sleep forever. So here, you can see here, they have some sort of abnormal single strand repair and deficiency. Just think about a situation where the PARP, we did the PARP inhibitor, right? And they have the single strand break, and then where the PARP is playing important role. So what will happen during the replication? These single strand break will eventually convert to the double strand break, but they have the efficient repair system. So this is called the HR, or double strand break repair system. So they can repair. So nothing happened to the cancer cells. Come up with the situations where they have the BRCA2. If you don't have BRCA2, then what will happen? You don't have HR, homologous recombination. So there will be like double strand break, but still they have some alternative energy pathway, Or if they cannot repair, they can go for cell death, apoptosis, but still they can survive. So this is not foolproof. But the third condition is like you don't have, you inhibit the PARP, at the same time, the cells or cancers, they don't have BRCA. So what will happen? This is a double blow. So that in that conditions, cell has no choice but to go for the death. So this process is given the name synthetic lethality. So what is the synthetic lethality? So you have normal cells, right? Normal cells having the BRCA2, and then you inhibit the PARP. So what will happen? You don't have those single strand break will not be happen, but still they have the backup pathway and the homologous recombination pathway, they will survive. But if you have the BRCA1 or BRCA2 deficient tumor or incompetent cells, what will happen? You inhibit the PARP, you don't have single strand break. Then inhibit the HR, you don't have singles. What will happen? The cells has no choice, they will die. This is called synthetic lethality. There's one gene or one is already there, and then you hammer another one, and then cells become so susceptible. So this is the one of the breakthrough in the cancer treatment, and then thousands and thousands of the cancer patients around the world have been taking this medicine, and mainly the credit goes to my boss, Professor Thomas Halliday. Back in 2005, he has discovered, and definitely, along with him, there are a lot of different people might have done the contemporary at the same time, but he was one who was outsmart, and then he published the paper in Nature in 2005 to say that with the PARP inhibitor, they can kill the tumor cells without inducing more toxicity. That means it is synthetic lethal to the cancer cells. And they are specifically killing the cancer cells. They are not harming the normal cells. So what he has done, like you can see, these are there are different kind of cell lines that they have complemented the same cell lines VB8, 9, and then they have complemented with this one. You can see that these are the BRCA2 deficient cell lines. They need just tiny amount of drug to get killed. But however, these different cell lines which has not BRCA, they are not sensitive. So you can see this very huge therapeutic window between the BRCA deficient and the BRCA proficient. Similar, this is the two different cell lines. And here you can see this is the MCF7 cell lines, and here you can see those are the BRCA2 deficient, and they are very, very sensitive, while the BRCA proficient or cells which has the BRCA, they are not at all toxic to the PARP inhibitor, similar to both the cell lines. So that is, back in 2005, it is called, you can say it is the target therapy, or you can say the synthetic lethal therapy. Then what they have done, definitely because they have seen in the cells, so they want to translate whether it was true or not. So then this is the VC8 cells are there, this is the control, and this is treated. But what we have done, they have the complemented with the BRCA. So once they have the complemented with BRCA, then they are not sensitive to the either like control, or you have treated with AG136. So they are not so sensitive to the cells. But if you have these cells VC8, which is BRCA mutant, or they don't have, so then they become very sensitive, and then there's no question that even they can regrow it. So after his discovery, there are so many labs, and they keep on working. They work a lot, but I'm not going to show anything beyond that one. And there are so many companies and so many labs, they have been working on the PARP inhibitor, how they can improve this PARP inhibitor. They use monotherapy, they use in the combination therapy, and then eventually they are getting a lot of success. Finally, in 2014, 13, 14, this PARP inhibitor is called olaparib. Actually, this is like my boss. He has the single patent holder from the University of Sheffield. From that, they have given to the kudos, and kudos sold out to AstraZeneca. And then these AstraZeneca, they gave lonzaparib or olaparib, and these are FDA approval back in 2014. They have used either as monotherapy or with bicazimib, that is the VEGFR inhibitors. So why they got approval? So they got approval because they have done solo trials on the BRCA2 deficient, and you can see the huge difference between the placebo control as well as the olaparib. So they have done that, and then there is increase in the progression-free survival. And what they have done, they have also combined the olaparib with the VEGFR inhibitor, and then they can see that even you have the much better effect compared to the VEGFR emitter. So that olaparib, they do have potential to combine with the olaparib and the VEGF inhibitor. But sorry, I'm getting some sound. And so this is the basis why they got approval. So what is the efficacy from the solo trial or from the polar trial? So here you can see that, and these one should remember, one should know that if you can take the PARP inhibitor when you have the BRCA deficient tumor, otherwise it will not work. And then there are a lot of patients who has taken cisplatin or first line of therapy, they don't respond it. Then when they take PARP, olaparib, they have very good effect. Like you can see that tumor can get vanish. Within 2 or 3 months of the treatment. So, but the problem was that they have there's difference between only 13.8 amines because of the development of the resistance. And then when they have used polar trials with the olaparib alone or olaparib with in combinations, they have a little better progression-free survival. So these stories, my boss has started this story in 2002, 2001, and they got approval in 2014. So one can think that it took almost 10, 12 years to bring the drug into the market. So what is the use of the olaparib now? So people have been using the olaparib in advanced ovarian cancers, they have been using in the breast cancers, and they have pancreatic cancers, metastatic prostate cancers. But all cancers have one thing in common, that they have some sort of somatic or germline mutation in the BRCA gene, so that you need to have the HR deficient tumor. If you have HR deficient tumor, then the likely to get benefit from PARP is much better. Otherwise, the PARP is useless. So with this one, I can say that, this is a lot of involvement, lot of research, and lot of money. And currently, more than every year, around 1 to 2 billion of the cost of drug selling throughout the world. So that was the first story, and this is another story. I'm going to talk about the Mth1, it is the Nudix family protein, and I am the witness to this protein because I started. I was the part of this project. And then when they started in 2010, I enjoyed the lab in 2012. So when I went, they were a little bit advanced. They already had validated the siRNA and other stuff. And then from there, I'm the witness to development of this target. So what is the Mth1 protein? So this is belongs to the the Newdix hydrolase protein. This is like nucleoside diphosphatase linked to moiety X. So these are the cleaner, they sweep or they clean. So what they clean? They clean the dirty pools. They clean the dirty pools of nucleotide pools. And these they're different, more than 22 types of Newdix protein. They have the common Newdix domain. That's why they came in one family. Even they say that Mth1 or not one or not two, but still they have different functions. Definitely, I mean, because and lot many has not been characterized. A few has characterized like an Mth1 or not 1, not 5, not 18. So our lab is also not 15, not 19. So we have been, and not 22. So we have the different Newdix protein which has been characterized and known to have some role in the cancer development. That's why. So what they do, and this is like these drugs, these protein, they prevent the misincorporation of the oxidized nucleotide to the genome. And this is one of the, we can say this protein is Mth1, and because by the hydrolysis. So this is like, they act as a phosphatase. So in the normal process, we have the ROS. This is common to get from the free radicals. They form because there is always the balance between the oxidative stress and antioxidants. If the balance got tilted, so they will have a lot of so there are a lot of free radicals, and then that means eventually there's increase in the reactive oxygen species. And then what they do the oxidation. They can do the oxidation to the lipid, the protein, to the genomic DNA. And then here what is happening, the pool, the DNTP pool, they are much more vulnerable to the oxidative stress than to the DNA. And here the Mth1, they play important role. So what they do, if there is some sort of oxidation, and there are different oxidized form of guanine or adenine, which due to the formation of the ROS, which has been taken care by the Mth1. And then one of the lesions is most of people might have aware, this is called the 8-oxoguanine or 2-hydroxyamine adenine. So these are the two main, but along with that one, there are different modifications of the nucleotides, which is being taken care by the Mth1. So broadly here, you can see that, the Mth1, and they work in conjunction with the different DNA glycosylages. It's called OGG1, or you can call the Mth1. So what they do, how they do. So just think for a scenario that you have the guanine, right? And then you have the ROS. So what will happen, like in the pool, because I say that it is vulnerable. So what they form the 8-oxoguanine, 8-oxo-DGTP. And this 8-oxo-DGTP, they can have the potential to, they can get along with cytosine as well as with adenine. So what will happen in the next one, there will be next applications, there will be A to T transitions and G to C transitions. So in general, when they have the oxidized guanine, it has to be taken care by the OGG1, which remove the oxidized, then they induce the ABS6 sites and they get repaired. And then adenine has been taken care by the Mth1. If they cannot do it, so they will induce the single strand break and eventually double strand break. So here the Mth1 is act like a sanitizer. They sanitize the pool. So why Mth1 is a good target? So you can see that there are different kind of cancers, ALL, lymphoma, sarcoma, testicular cancers. the expression level of these in these tumors, they are much more higher than the healthy tumors. And here you can see also the protein level in the different protein levels, right? You can see that, in the colorectal cancers, they have a very strong presence of the MTH1, in the breast cancer and testicle cancers. So the expression level is not the same. So it is likely that, and we have seen that MTH1 does not work in all the cancers, but it does work in many cancers. So as I mentioned that how we start with the validation of the target, right? So we use siRNA, and then definitely we are showing 1 to 3 siRNA, because indeed this is so difficult process, because you buy the siRNA, it doesn't work. So we need to take such siRNA, and it takes a lot of time to validation. But here you can see that when you deplete with the siRNA, the MTH1, there is a difference of clonal survival. Means cells become very sensitive. Cells have become sensitive. They survive less. And then if you inhibit the MTH1, if you deplete MTH1, what will happen? They trigger the repair process. Here you can see there's phosphorylation of ATM or the P53, these and the P21. So that means somehow the MTH1 depletion triggered the DNA damage. And because it is preventing the incorporation of atoxoguanine, so think a scenario when you don't have the MTH1, then what will happen? They're supposed to have the more integration of atoxoguanine in the DNA. But here you can see that with just for the siRNA there is decrease. Then when we complement it with the wild type MTH1, we can see the rescue the effect. And then we have the catalytic death, we don't see the risk. That means we are on the target, and it is mainly happening due to the catalytic domain of the MTH1. And then these are happening only in U2S cells, and these are the VH cells, VH10 cells. These are the forescreen fibroblasts, so they have no effect. So that shows that these target is mainly cancer specific. And then even you see the damages, the 53BP1, these are the marker for the double strand. You can see that when you have the siRNA and the non-targeting, they don't have much damage. When you have siRNA or deplete the MTH1, you have the massive increase in the 53BP1, and then which can be rescued by the wild type. You put back the MTH1 and they become fine, but not with the catalytic day. So these what they do. So here with this we can say yes, we have validated the target. And then definitely here the similar one, you can see that they have increase in the atoxoguanine in the siRNA compared to the wild type. So what they do, that one can say that MTH1 prevent the atoxoguanine incorporation and required for the cancer cells. So one thing I want to introduce that, I'm sure everybody might be aware of the Comet assay, but here we use the modified Comet assay, because what we did, like when you treat the cells, when you deplete the MTH1, they will have a lot of atoxoguanine incorporated in the DNA. And then when you add the enzyme, the OGG1 enzyme or Mth1 enzyme, these are enzyme I just mentioned about the 2-hydroxyadenine or atoxoguanine. So it has been taken care of by these two enzymes. So when you add externally, so they induce a lot of damages. So you can see that in the non-targeting, or they don't have any effect, but with the MTH1 depleted, with the OGG1 and the Mth1, you see the massive effect. So, and then we also have done, V signaling pathway. And so they have involvement of the ATM, they have the, which is involved in the homologous recombination. We have the RAD51, which is also the part of homologous recombination, and DNA PK. So this is not that, we cannot say that MTH1 process is repaired by the homologous recombination or NHJ pathway or the BR pathway. Like it has a lot different functions it has inducing. So, but however, definitely we can see that with the catalytic domain, we can rescue the effect. And then what is happening in vivo? So all we did with the shRNA, and then what we do, like we try to validate in the mouse system. So what we have done, like we have here the non-targeting, and these are the doxycycline inducible system. So when you add the doxycycline, they deplete the MTH1, and then you can see this one, when you add the doxycycline after like 10-15 days. So once the expression goes down, the tumor got to decrease, and they touch almost bottom. And here in the figure panel M, you can see that the mice, they survived quite long time. Almost no mice died. Which got the doxycycline. That means these are they're working very good. And how they work? Here you can see that with the siRNA, they induce the apoptosis. So this is, as I mentioned, that they have the process. So because the MTH1, they induce some damage, oxidative damage, they might be taken care by the ATM or RAD51, but somehow they cannot get repaired, or they have the more than 40 lesions. They eventually send the signals and cells got died. So then what they did, okay, they thought, oh, this is the very good target. And then what they did, like they purify the protein. That is the Mth1. This is the homolog of the bacterial homolog of the MTH1. And then we have collaboration with the crystal structure. And then eventually they have the 4 different compounds, and they make the activity assay. You can see here the scaffold 1, which has like activity of 700. They don't have much effect, but other 3 inhibitors, they inhibit the activity of the DGDP activity. And that was the starting point of the development of MTH1 inhibitor. And then they have made in the beginning for the proof of concept. So I have the 3 different kind of emitter, as they gave name. TH means Thomas Halliday. It's like a TS287, 5F. You can see here by this time we had made almost 600 compounds or more, and then we got only one hit, which has been which can be workable, or which can use for the proof of principle. And because they inhibit, and what we need, like we also need the drug to be very potent. See, in the in vitro condition, at the just for 5 nanomolar, they can stop the activity, IC50 well. And then here, how we validate. So this is one of the, the panel B is we call the set size, called the cellular cellular thermal stability assay. This is the principle that, like some protein, when they have they have at certain temperature, they melt away. So this is like, they melt away, the MTH protein, they melt away. But when they have some drug is interact with the protein, the stability goes more. So this is the way you can say that the target engagement. That means these drugs, they bind with the protein and they inhibit the activity. Then panel C, you can see that these white, and they have VH10 cells, SD and HP. So these are the cells which is not cancer cells. So you can see there's a huge difference there's a huge therapeutic window, or like there's a remarkable difference between the normal cells versus. And then we took the different kind. This is the osteosarcoma cancer cell lines. This is the breast cancer cell. This is the breast cancer cell lines. This is the colon cancer cell. So these cell lines being selected based on the expression level in certain protein. And then similar to that, what we seen with the siRNA, we see the similar effect. When you treat with MTH1, you have the high tail moment. High tail moment after the addition of OGG1, and they denote that these drugs, they induce oxidative stress or oxidative damage. And for this one, we can also use the positive control, the H2O2, which induce atoxoguanine, and then which has been, that's why you have the very long tail moment. And here, and this is the quantification of the 588. You can see that this is after IgG1, you have the more damage compared to the control. And similar to the siRNA, we also did 53pp1, and then you see that 650, they don't have MTH1 inhibiting activity, so they don't induce so much. But 287, 588, they induce DNA damage. So the drug and the target is the same. And what we have done, then we have used different cell lines. You can see here the 287, 588, and 650. They have the different the different cell lines being treated with the 588 or MTH1 inhibitor. You can see the IC50 value is like more than 20 in most of the normal cell lines, where these cancer cell lines of the different origins, lung cancer, prostate cancer, colon cancer, breast cancer, so almost so many cancer cell lines, and these are RAS-induced cell lines, the fibroblasts. So these are the very sensitive to the MTH1. So they strengthen our belief that we are on the right track. And then what we have done, like definitely, as we know the process, the proof of principle, proof of the drugs has to be taken care to the preclinical, because our goal from our lab, the goal is to have a treatment. We don't believe in we believe in product. We do believe in paper, but our main concern that we can give something to the society. So we didn't stop there. Then we had the competence here in the lab itself, and then we did the Cmax, we did a PK, and then based on the PK, we choose the drug based on the Cmax or the T half, and then you can see there is a difference in the mice treated with the 5888 or mice treated with the vehicle, and then without any toxicity. So these drugs, they don't create any toxicity. And then this is like, the colon cancer xenograft. And then we also use the MCF, different lung cancer, the breast cancer. Then you can see there is a difference. Okay, but not so much, but they don't have any toxicity. So these reform our thought and process, and then we keep continuing. And then we also checked for different parameters here, right? And then there is no difference between the other vehicle treated or the MTH1. So these, we can say that these drugs is not at all toxic. Eventually, I was a little late, and then to join this lab by the time they have done, and then I contributed this paper, and then they published in Nature. But once they got published in Nature, there are many companies, many labs, they jumped into this project. And then we have some questions that some of the labs, some of the company, they could not reproduce their own. They could manage to have their drug MTH1 inhibitor, but they didn't find the similar result. Then we did the reinvestigation. So as I mentioned, it is very difficult process. So we have used atoxoguanine measurement. In this time, we used immunofluorescence or confocal microscopy. And then what we did, like we used potassium bromide as positive control, and then we saw with our drug. So our drug definitely they induce atoxaguanine, but the drug from AstraZeneca, they did not induce, or so from Bayer, so they failed to induce some sort of damage. So here we can say that. And then we have also the correlation with the enzyme activity, the non-viable cells, and CETSA. So we found that we have very good correlation, that if you have the high enzyme activity, inhibition, and then definitely the cells are less survivable, and then you have the good engagement. But same thing that we did not find with the other inhibitors. And then we presume that the cells, the drug which did not induce atoxaguanine, they don't kill the cells, cancer cells. So, and then we use for the lead optimization, what we did, like we further moved from 588 to different drug, and here we use 1579 because 588 has the not very good round of PK parameters, but here with we use the 1579, and then we also try to rescue. You can see that with the empty vector, we see good tail moment, but with the Mth1, it means bacterial homologue, we could rescue the effect. So that we wanted to say that we could rescue the effect. That means even with the different inhibitor, same inhibitor, different series of inhibitor, we are on target. And then we can see that with the Mth1 overexpression, we can rescue the effect. Even with the NAC, it means NAC is this is the Ross scavenger, and then we can also see some sort of rescue effect. And with the MTH1, we see the rescue effect. So that means that with the 1579, we are still on the target. And then we have the difference between the normal cells and the cancer cell. You can see almost 9-fold difference in the IC50 value of these two. And then it also correlates with the high enzyme activity, the CETSA, and the non-viable cells. And definitely with the different concentration, different micromolar concentrations, they induce the foci. And similar to 588 or similar to MTH1 depletion, they induce different damage, like they induce the phosphorylation of P53, they induce the phosphorylation of, they induce the P21, they induce cleavage PARP, cleavage 3, and they induce the damage. So all together, one can conclude, one can say that we are, and the 1579 is the different, means the similar to the 588. And then, no, because when you're moving to the clinic, so this is the big risk. Big risk of what? So we don't know if these drugs can also target the different, like the relatives. So they check the activity. So 1579, they have just activity against MTH1, but they don't inhibit other different nutics. So that means these our inhibitors, what we have made, is also on target. And then this is one of the classical and very expensive assay to carry out. It's called the Proteomics EPSA. This is also one of the way to validate the MT1 TPP and target protein profiling. So this is one of the way with the mass spec and it's called alpha screening. One can determine and the specificity of the drug. And then here you can see that we have the about 5,000 protein, and then here the MTH1 was outsmart with the 1579. And then similar with the other, with the CETSA, you can see that there's a huge difference, in the target engagement. So either live cells or cell lysate or TPP, this all denotes that there's very great target engagement, or the of the 1579, which goes to the MTH1 and then inhibit the NPH1. And then and similar to the 588, we used you also use the mice, and then this time we also compared with the 5FU. We compared with one dose versus two dose. We compared with three different doses. It is very important to see whether we do seems if there is effect. So there should be effect based on the dose. So we do see the dose dependent response. We do see single dose versus double dose, and then we see that how our drug is compared to the existing drug in the market. So we here compared, it was the 5FU is one of the drug of choice for the colon cancer, and then you see that only the 5FU it has some problem with the toxicity, but not with the 1579. So the same thing with the similar to the 588. These they also strengthen or cementing the effect. And then we also use some of the PTX model. This is the xenograph from the patient-derived melanoma cells. Then you can see massive effect with this in the mice. And then definitely we also tested, so far we had tested adeoxyguanine in the DNA in the cells. Here we check the adeoxyguanine level in the tumor, treated with 1579. Then you see this very high level of the adeoxyguanine. And then you have the different damages of 50 BP1 foci, the gamma S2X foci. This is cleavage space 3, or on Cas61, this is the proliferation marker. Then you can see that there is decrease in the proliferation once you treat with 1579. But however, why decrease? Because there is a lot of damage induced by 1579, and that leads to the replication stress, or the cells are not dividing. Eventually they got they could not divide. And here we have three different mice numbers, and you see there is the induction of the phosphor form of P53 gamma S2X cleavage carpus. So these all suggest that our drug, they induce DNA damage, they induce adeoxyguanine. Through that, they have some sort of toxicity. So then we published in Annals of Oncology, which is one of the high impact factor journal in the cancer research. So, but still, people were not satisfied, and we were not satisfied that why the others are not getting, then we are getting the data. So what we did, then our one of the colleague, he did some Mth1 for long-term effect. So what he found, that this Mth1 is not only inducing the adeoxyguanine, they also have some sort of polynucleation or micronuclei formation in the cancer cells, similar to with siRNA or in the shRNA. So they have the problem with the anaphase with the shRNA, toxin-induced with shRNA. And when you do with the siRNA, you have the prolonged mitotic phase. That means they need more time. There is some sort of aberration happening in the mitotic process. So they cannot, they cannot go to the next stage. And then they also use the CREST and with the siRNA, and then they can see that what they found, that the interkinetic core distance, they become shorter in the Mth1. This is Mth1 protein. So we believe that that Mth1 do have the role in mitosis. And then similar to the siRNA, we used both the drug 588 and 1579. Then we found a similar effect, that they do the polynucleation, and then they do the macronucleation effect. And then there is increase in the time in mitosis. So they suggest that maybe it is possible that some of our inhibitors, they might have also having some effect, or they have some role. It is not sure. It is the off-target effect or the on effect, but they have some role in the mitosis. So, and then you have the RAS. So you can see that they have the quite a lot of damage. It's called the DIM, and death-induced mitosis is more with the our MTH1 inhibitor compared to the DIM. So with this, we believe that we are on target, but it's still hard to say. And then, we also use paclitaxel, which is one of the known mitotic poison. And then, we do see significant difference in the intercanticle distance between, but we don't find significant difference with the paclitaxel. So we thought like it could be, the different mechanism of action, but somehow they bring out some effect on the mitosis. So what kind of effect on mitosis? So what we found that either you deplete MTH1 siRNA, and so they have some sort of problem in microtubule polymerizations. MTH1 protein itself they do, and then if the MTH1 protein does a similar, we also see with our MTH1 inhibitor. So we still believe that we are on target, but people do believe that it is off target effect. So what we did, to overcome the black clouds, we use our inhibitor, then we use also AstraZeneca's inhibitors. So what we found that we don't have much effect, but when you have atoxoguanine, when you supplement atoxoguanine, you see the massive damage. Even with the these drugs, they don't inhibit the polymerization. So we can say that in order to kill the cancer cells, the drug should have both the effect. They should inhibit the MTH1 as well as they should induce ROS, then atoxoguanine, atoxo-DGTP formations. So here, these are like they have done in the zebrafish embryo. And then we modify our hypothesis, and then what we think that it has the both mitotic inhibitor or MTH1, they do some sort of mitosis, and it is known that during the mitosis there is a surge of ROS formations, and then these ROS formations they will definitely have a lot of impact, and they make the pool very dirty. There they need our MTH1. Once they need MTH1, if they don't have MTH1, they will incorporate it and they can go to a cell death. And then how we have proved that one. So we have the AstraZeneca's inhibitor and then paclitaxel. So these are the mitotic poison. These are MTH1, pure MTH1. They don't inhibit the ATAL, they don't inhibit the polymerization. But when you combine it, you see the massive damage. So these images or these results, they suggest that what the drug we have is the dual inhibitor, because we also see some sort of synergy when you combine paclitaxel with the AstraZeneca's inhibitor. And eventually our drug, is already, we had, after that we also use a lot of different cell lines. We used various mouse models. We have used patient-derived xenografts to strengthen our belief. Then we have done a lot of panels. We have done preclinical studies in the dog. We have the safety studies. Then finally, we think that, oh, this is one of the clinical candidate, and then we should test in the clinical trial. So in 2017, we have started in the phase 1 clinical trial in the different solid cancer. The objective was to evaluate the safety and tolerability of the current and then to find the maximum tolerant dose. Then along with that one, we have another clinical trial going on with current in the Mth1 clinical trial, and objective is also the same. And then we want to see whether, if we can go in advance in the phase. And so far we cannot disclose anything, but so far we think, soon we will produce our results. So far so good. But to come up at this stage is almost 10 years now, 10 to 11 years. And then with this many people been working in this project. So I can say thank you so much for your kind attention. I'm done with my presentation. If any questions, please feel free to ask me.
Thank you, Dr. Kumar Sanjeev, sir. Now the participants can ask the question. Any question? There is no question, sir, in the chat box even. So I thank you, sir, that you accept our invitation and give a presentation here and share insight over the inhibitors that can be used against the cancers. And as you told that you are working on a novel inhibitors, novel drug that can be, we can expect that you will give that drug up to next 3 to 4 years. So
I hope so.
Yeah, we may have a new drug again that can treat the cancer.
Yes, we hope so.
So thank you, sir. Thank you very much.
Thank you so much.
Okay, thank you. Thank you all the participants. So it's all over for today. We will meet tomorrow at 10:30. Till then, bye. Take care.